Hello everyone, I will be going through the topical exercise for mitosis and meiosis. This is a rather long RE, so you can skip to the questions where you made mistakes in and you want to learn from. Okay, let's begin with question 1 and 2. In question 1, you'll find a graph, and this is a very typical graph that we like to provide for this particular chapter. So I would like you to star this question. Maybe you want to include this graph in your notes. Okay, usually the x-axis will show the amount of DNA, and the starting point is usually referring to a regular cell with diploid number of chromosomes. That means the original number of chromosomes. So in the case of human beings, you expect to find 46 chromosomes at the starting point, right? And so that means to say, at this point over here, there is twice the number of DNA, right? And that means at the lowest points, there is half the number of DNA. So that means at the lowest point, we expect 32, and on the top, you have 92. All right, so that's how it goes. So let's try to make sense of this graph. Which option indicates the complete process of nuclear division that is used to produce gametes? What are gametes? Gametes are sex cells, which in other words refer to like the sperm or the egg. And the process that results in sex cells is meiosis. That means to say we have to find a part of the graph that represents meiosis. Now, how do I tell? Here, if we see over here, the amount of DNA seems to double. After doubling, it remains the same, and then there's this drop, right? But you see, before and after the drop, you have the same amount of DNA, 46 before, 46 after. It means to say that whatever happened in between has to be mitosis, because mitosis results in the same amount of DNA before and after. Just to be very specific, actually, this portion over here where the amount of DNA is doubling, you know what that phase that is? It is the S phase, because that's when DNA replication occurs. So actually, it is in this plateau part. This plateau part is actually when mitosis occurs. Notice the amount of DNA doesn't change. Okay, this drop over here represents cytokinesis, because this is when the cell divides into two, and then we can safely say that the amount of DNA will change. All right, so take note of that. So when does meiosis occur? Well, if we look at the second graph on the later part, S phase again, double the amount of DNA, then meiosis occurs. At the end of meiosis, you expect a drop, but this time to half the amount of DNA. And that means this segment over here is likely to be meiosis, since it results in half the amount of DNA. Okay, and therefore, the phase we are looking at is between 3 to 5, and therefore the answer is C. Look at question 2. At which phase of cell cycle do the processes 1 and 3 occur? 1 is this slope over here, and 3 is also this slope over here. What results in doubling the amount of DNA? Well, it has to be S phase. This is when DNA replication occurs. Alright, so the answer is D. Now let's go to the next question. The next question is question 3. It says that this particular creature, this is actually a mosquito, okay, Aedes aegypti, are diploid organisms, and then they put 2n equals to 6. You may be wondering what n represents. Okay, n represents the number of chromosomes in one set. What does this mean? I'll give you an example. Let's humans have 46 chromosomes. So actually, we have 2 sets of chromosomes, with each set n being 23. Right? N refers to the number of chromosomes in one set. So for human beings, in one set we have 23 chromosomes. In 2 sets, we have 46 chromosomes. And we sometimes say 2n refers to the diploid number and n equals to the haploid number. Right? So don't be surprised. If I say, what is the haploid number of chromosomes for humans? You can say 23. If I say, what's the n number of chromosomes for humans? It's also 23. What is the diploid number of chromosomes for humans? 46. What is 2n? Number of chromosomes is 46. Okay? So, which of the following is a false representation of the number of chromosomes and DNA content of the respective stage in a cell? Do take note, number of chromosomes and DNA content, actually they are two separate things. Why? Okay, I'll give you an example. Let's say in S phase, I have one chromosome, right? So here I have one chromosome. I'm going to write one chromosome. After S phase, right, we now have still one chromosome, right? It's two sister chromatids attached at the centromere, and it's still one chromosome. However, has the amount of DNA changed? Yes. The amount of DNA before S phase is, let's say, 1. After S phase, we now have 2 times the amount of DNA, except that the DNA is attached to each other at centromere, and therefore we still have only one chromosome. Okay, so there are two separate things. Please take note. This is a very interesting question. You may want to star it. Right, so which one? It gives a false answer. Actually, number 1, A is the false answer. Okay, why? At anaphase, the number of chromosomes is 6. The amount of DNA molecules is 12. So let's try this. I'm going to draw out the number of chromosomes. Okay, they say that a diploid organism has 6 chromosomes. So let's say I'm going to draw in a cell right now. All right, 1, 2, 3, 4, 5, and 6 chromosomes. This is during metaphase, right? During anaphase, what happens? The chromosomes will separate, right? Each of the chromosomes will separate into daughter chromosomes. So how many chromosomes do we have in total if we were to give it a count? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Right? We have actually 12 chromosomes, not 6 chromosomes at anaphase. Okay, that's why the answer is A. Moving on to the next question. Okay, for this particular chapter, we really like to show you diagrams or pictures of real cell division taking place. And the skill to pick up here is to be able to identify what phase. Right, here they show you a few cells. They want you to identify the stage of nuclear division it is undergoing. And the answer is D, prophase. But how do we tell? I would like to give you some tips on how to identify cells in their various stages, just based on photograph. Okay, because you'll find that the diagram versus the photograph is a very different thing, right? Okay, so let's have a look. Number 1, how do we identify cells in interphase? First thing we know is that in interphase, the nuclear envelope still exists. Okay? And secondly, the DNA has not condensed. What does this imply? It implies that in real life, if we were to look at a real cell in interphase, you'll find that the nuclear envelope, the shape, this circle shape will be very intact. And number 2, you actually won't see thick squiggly worms. Alright? Every time you see thick squiggly worms, it means to say condensation has occurred. But you look at this particular nucleus over here, instead we see a lot of like dots here and there. When you see dots, right, it means that the DNA has not condensed yet. Whereas if we look down here and here, you can see very squiggly worms, right? These squiggly worms means that the DNA has condensed. Okay, so this is how I identify interphase. Number 1, presence of nuclear envelope, and number 2, has not condensed yet, or no worms. Okay, that's how I identify it. Number 2, how do we identify prophases? In prophase, one of the things you have heard is that the nuclear envelope starts to disintegrate. Now we look at this over here and this over here. Notice that the nuclear envelope no longer exists. Yeah, but the true sign of prophase occurring is the presence of this thick, very distinct worm-like structures. Those are condensed chromosomes. Okay, so I will identify prophase with the presence of condensed chromosomes in the form of like thick worms. Okay, and in fact in late prophase, notice you cannot even see the nuclear envelope anymore. You don't see any circle at all. The worms are kind of like dispersing already. Next, mitotic metaphase. In metaphase, we know that the chromosomes align singly at the metaphase plate. Now, if you look at the diagram on the left-hand side, it doesn't quite look like what we see in the diagram that we are very used to. But this is the best you can, if you get used to it, this is the best we can see of chromosomes aligning in the middle. You can sort of see that this is the metaphase plate. All the chromosomes seems to be aligning in this fashion. Another clue also is that you don't see the chromosomes moving apart. Remember, if they're moving apart, you will see them in this formation, right? Kind of moving towards opposite ends. But here, we find that they are still kind of pointing towards the middle, right? So if you see chromosomes still pointing towards the middle, like what you see over here, it means that they are all in metaphase. But if you see them pointing away, that means we are going to anaphase. Here you can see all the chromosomes pointing away, right? You can see, right, roughly that they are pointing away. Right, so that's a clear sign that it's in anaphase. And finally, telophase. Telophase, you find that the chromosomes start to be in opposite ends of the cell. And usually by this point, you'll find that the chromosomes are very densely packed with each other. And notice that the worms are no longer that visible. That's because they are also decondensing the DNA. Okay, so these are some tips of how to recognize the various phases based on images provided to you. So let's look over here. What phase is this in? Okay, I would, number one, use the tips. Okay, first and foremost, notice that nuclear envelope not so present anymore. I can tell nuclear envelope is still present if you can see very nice, circles like these all surrounding. So actually all of these are in interphase. But the clear sign is that we can see all these worms, right? These worms means that they are condensed DNA. Okay, very condensed DNA. So that's why it's prophase. In fact, this one is also in prophase. But the rest of them, I would say they are still in interphase because we don't see any distinct worms. Okay, please don't use the word worms when you're doing your questions. Just using worms so that it's easier for us to recognize. So now question 5 also needs to make use of the skill of identifying the phases. Let's try. Which one is likely interphase? Number 3. Number 3 is likely interphase. Why? Nuclear envelope still present. Notice the chromosomes have not condensed. Still exists as a lot of dots. Number 4 is likely prophase because you can see the worm-like structures exist. Those are the chromosomes, condensed chromosomes. This is likely metaphase because you can see all the chromosomes pointing inwards. And there seems to be a metaphase plate that they're pointing inwards towards. This one is likely anaphase because you can see them pointing away. Finally, this is telophase because you see them in opposite poles. And also you cannot really see the worm-like structures anymore. So what's the sequence we want to go by? We will go by number 3 as the first one, followed by number 4. Okay, after that we'll go into metaphase, and then we will go into anaphase, and then we'll go into telophase. And therefore the answer is D. Okay, it was quite fun to identify once you know how. Question 6. The second division of meiosis differs from mitosis because in meiosis, second division of meiosis differs from mitosis. Actually, the second division of mitosis is very similar to mitosis, right? Because in the second division of meiosis, all of the chromosomes also line up singly at the metaphase plate. Okay, so what is the difference? Answer is D. The separating chromatids differ genetically. What does it mean? It means that, okay, if I were to look at a single chromosome, they are saying that the chromatids that are separating apart, okay, I'm drawing them separating, pulling to the opposite poles, they are not genetically identical. Is that true? Okay, meaning that we say that it's not genetically identical. So if I put capital A means that it is genetically identical, right? But what they are saying is that it is not genetically identical. Is it possible? Yes, it is possible after crossing over. And crossing over only takes place in prophase 1 of meiosis 1. Yeah, so that is one of the key differences, that the separating chromosomes, they need not be genetically identical. Whereas in mitosis, they will be genetically identical. Okay, why would they be genetically identical? Because they just went through S phase. Whatever you find will be exactly the same, right? They are twins, they are conjoined to each other, okay, after S phase. So before S phase, it will look something like that, right? Then after S phase, it will look something like that, okay? But in meiosis, what will happen is that if there is a crossing over, so let me draw another homologous chromosome. Okay, maybe it is exactly the same, but maybe here is small b. If there is crossing over, then it can result in this over here. Okay, so that's question 6. Next, which statement correctly describes homologous chromosomes? The answer here is D, right? They carry the same gene loci. Gene loci means like the location of the gene. So that means to say, here I have 2 chromosomes. What makes them homologous chromosomes is because they carry genes at the same locations. So here, although the alleles are different, the location of the gene is the same. So that's what they mean by carry the same gene loci. Okay, location of the gene is the same, both B and in the same location, but the alleles can be different. Okay, that implies that these are all wrong, right? Okay, so is A wrong? They are formed during meiosis. No, homologous chromosomes are not formed during meiosis. B, they are genetically identical. Not necessarily. Remember, crossing over can take place, which will result in them not being genetically identical. They are held together by centromeres. Actually, this is correct, but it is also correct, but it's not what defines homologous chromosomes. Yeah, because I could have another chromosome that is maybe much smaller, but it's also joined together at centromeres. But these 2 chromosomes aren't homologous, even though they both have centromeres. So homologous chromosomes, that's not what defines homologous chromosomes. Another way to interpret this, it could mean that they are saying that the homologous chromosomes are joined together at the centromere. It's very weird. We don't see 2 butterflies joined together at the centromere either. Okay, so D is the best answer. Next, we go to question 8. It shows this diagram. They ask you what phase of meiosis is taking place. Immediately, I would say that this is anaphase 1. How do I know it's anaphase 1? Because if we see over here, what is separating are homologous chromosomes. That means right before this phase, we had homologous chromosomes that are lined up in bivalence at the metaphase plate. Right? That means this has to be metaphase 1. It is not metaphase 2. So I would say this is likely anaphase 1 taking place. Also another clue, notice that crossing over has taken place. Okay, shown by the different colors. Crossing over will have taken place in prophase 1. That means this is not anaphase 2, it's anaphase 1. What will anaphase 2 look like actually? Anaphase 2 will look like this. It will look like metaphase. It will just look like mitosis, right? The anaphase of mitosis, where you find sister chromatids being pulled apart, right? So anaphase 2 will look like this, and it also looks like the anaphase of mitosis. So the best answer is A, and that's why I gave this little fun activity to ask you, what phase do you think this particular chromosome in our atrium looks like? Okay, it's actually in anaphase 1, right? Because we see an intact chromosome with 2 sister chromatids. Okay, so that was just a little fun activity for us. We are now done with the MCQ. Let's go to the structured question. I would say this structured question was quite well done. I'm very proud of all of you for being able to identify the phases. Right, if we practice using these images again, let's try again. Yeah. Okay, let's look at A. I would say A is telophase, right? Because we find that the chromosomes are now at opposite ends. Looks the same, right? What is B? I would actually say B, right? I'm not entirely sure. Because I can see distinct worms, I may say that B is something in a late prophase. What about C? C, I would say that this is likely post-cytokinesis, because I can see 2 distinct cells. What about D? This is a tough one, right? We see that things are kind of aggregating in the middle. So actually, if things are concentrated in the middle, I would say the best bet we have here is there is metaphase, where things are concentrating in the middle. Okay, so what would the sequence be? I would say it would be B, which is prophase, followed by D, metaphase, followed by A, telophase, followed by C, which is cytokinesis. So that's why the order is BDAC. What is D? D is metaphase. And how do I know? Because the chromosomes line up singly at the metaphase plate. Okay, please take note of the word singly, because it will help you differentiate the metaphase in meiosis, right? In meiosis, metaphase 1, you'll find that the chromosomes don't line up singly, right? They line up as a pair in the form of bivalence at the metaphase plate. So the word singly is important. Moving on, this particular question, again, they want you to identify what phase it's been arrested at. Notice that the chromosomes kind of lined up, concentrate in the middle. So I would say this is metaphase. Okay? And suggest how vinblastine acts to prevent mitosis. When you see the word suggest, it means that this is an open-ended question. There are multiple answers. As long as you apply the right logic, we can award you the marks. So there are various answers. For what reason would the cells align at the metaphase plate and then stop being pulled apart to opposite ends? For several reasons, one can speculate. Maybe the chemical inhibits the formation of the mitotic spindle. If the mitotic spindle doesn't form well, then we cannot pull them apart to opposite ends, right? Maybe it inhibits the formation of kinetochore microtubules. If the kinetochore microtubules don't attach to the chromosomes, then we cannot pull them apart to opposite ends. Maybe it prevents binding of kinetochore microtubules to the centromere. If it doesn't hook on to the kinetochores, then it cannot be pulled apart also. Maybe the chemical prevents the polymerization of microtubules, means the elongation of microtubules. That's also possible, right? If it doesn't elongate, it won't be able to attach to the kinetochore. So many different answers. I will have given you a tick if your answer is logical. but these are the various suggestions. Okay, now let's go through part B. They ask you to explain why meiosis is necessary during the formation of sperm cells. I noticed that students either give 0.1 or 0.3, but not both. Okay, let's look at 0.1. Meiosis halves the amount of genetic material so that you get haploid sperm cells. Okay, why is this important? Okay, many of us were able to say that it halves the amount of genetic material, but we don't explain why it is important, why it is necessary. It is necessary for fertilization, because at fertilization, haploid sperm cell will fuse with a haploid egg cell, then you'll get a zygote that's diploid, right? So make that link to fertilization. The second point, meiosis introduced genetic variation in the resultant sperms, so that no two sperm is the same, right? And this is really important. Okay, and genetic variation occurs through two processes. One in crossing over, which happens during prophase 1. Another independent assortment of chromosomes, which occurs at metaphase 1. I'd like to elaborate on the two right now. Okay, so let's look at prophase 1. At prophase 1, you'll find that the homologous chromosomes line up at the metaphase plate. Here I have two pair of homologous chromosomes. Imagine if crossing over occurs between this point over here, alright? That means to say I'll end up getting small a and big A, right, if crossing over occurs. And that means to say, if we go through with meiosis, we'll get one combination of sperm cells that goes something like that. Big A, big B, small a, big B, big A, small b, small a, small b. Okay, this is one combination. What if crossing over also occurs between here, right? Let me swap things around. Maybe now it's small b and here's big B. Then actually we get a new combination of sperms, right? The first one will be the same, but now we get a different combination here. We get a different combination here, and we get a different combination, the same combination at the end. So crossing over helps increase the number of combinations of alleles in the sperm cells and the egg cells. This increases genetic variation. Now we have many different combinations of sperm cells, such that when it fuses with the egg, no two individuals will look alike. Okay, so crossing over increases genetic variation. Next, independent assortment. For those that read the textbook, they will have mentioned that this happens in metaphase 1. What is independent assortment? Okay, so at metaphase 1, here I've drawn two pairs of homologous chromosomes. This is pair number 1, pair number 2, the red and the blue. We look over here. They've aligned themselves in this way, and if I were to go through to the end of meiosis, you'll find that one cell will have the combination of big A, big B, big D, big E. A, B, D, E. One cell will have big A, small b, big D, small e. Okay, A, B, D, E, and so on and so forth, yeah? And the last one will be A, B, D, E, so A, B, D, E. I'll have these number of combination of sperms. What is independent assortment of chromosomes? It means to say this. Notice that let's look at pair number 2. Right now, this chromosome is on the left, this chromosome is on the right. But nothing is stopping the chromosome on the right from being on the left-hand side, right? So imagine now it's on the left-hand side. And the chromosome on the left is now on the right-hand side. Just from assorting differently, right, either being on the left or the right, it can result in very different combinations of chromosomes, right? So let's have a look now. Now one combination of chromosomes will be A, B, D, E, right? A, B, D, E, right? Another combination could be A, small b, small b, small d, small e. Notice that the combination of the alleles is starting to be very different, right, from the original, if you compare across. So this is what independent assortment means. Independent assortment means that how the homologous pair of chromosome number 2, how this pair arranges itself on the left or on the right is independent from how the pair of chromosome 1 arranges itself on the left or on the right. Right? So that's what it means by independent. Independent means how this pair arranges itself on the left and right is independent from how the pair number 1 will arrange itself on the left and right. But you notice whether they arrange themselves on the left or right affects the resulting combinations of alleles at the end of the day. Okay, that's what independent assortment means. Okay, so but this also helps to increase the genetic variation. Each sperm now becomes very different from each other because of how the chromosomes arrange themselves at the metaphase plate. Okay, now let's go to the final question. They ask you to state and describe the phenomenon that occurs in prophase 1 that allows for genetic variation. Okay, in prophase 1, the thing that occurs is crossing over between non-sister chromatids of homologous chromosomes. So I just want to reiterate. So here I have two chromosomes. These two are sisters to each other. However, these two are non-sisters, right, of homologous chromosomes. This is a homologous pair of chromosomes, right? So we refer to sisters. They are only sisters if they are joined together at the same centromere. They are not sisters if they are not joined to the same centromere. So it is crossing over occurring between non-sisters of homologous chromosomes. This allows for exchange of genetic material so that we can produce different sex cells or gametes. Okay, we're at the last question. So this particular question says that at the end of meiosis 2, somehow the daughter cell contains 42 chromosomes. It's a bit weird, right? We know that at the end of the day, the daughter cell should have n number of chromosomes, right? From 2n should become n. So means to say, if it starts off with 42, it should end up with the half number 21. But here, we still have 42. So what does this imply? Well, then the key question is, what causes the number of chromosomes to halve from 42 to 21? Well, it halves because of a few things. Number one, it halves because anaphase takes place, right? Anaphase helps to separate the homologous chromosomes or the sister chromatids apart from each other. And when we separate them apart, it helps us to bring the chromosomes to opposite ends of the cell. Okay, bringing them to opposite ends of the cell is important so that we can eventually sort them into two different cells. Number two, cytokinesis. This is also important so that we can help to divide whatever DNA we have into two. If the cell doesn't divide into two, then we may not divide the amount of DNA into each of the two cells. So what could be the possible cause that results in the same number of chromosomes before and after? Well, it could be that number one, the anaphase is affected in some way. Okay, anaphase may have been affected, causing the chromosomes not to separate into two separate cells. There may have been an issue there. Okay, then number two, maybe the cell didn't even divide into two. Right? Maybe it didn't divide into two. So everything still remained together. And if it remained together, it means that we will still have the same number of chromosomes before and after. Okay, so that's the answer for question 3. That there may have been a failure at anaphase, and therefore the chromosomes didn't separate into two separate cells. They all concentrate into one. Or there could be a failure in cytokinesis. Maybe the cell didn't even divide, so the DNA didn't get to be separated into two separate cells. Okay, so this is a suggest question. So again, it's quite open-ended. If it makes sense, could have accepted the answer. Okay, this was a rather long video, so I hope that you learned something from it. And do make sure you do your self-evaluation at the end, so that you know which sections you want to improve more on. Okay, thank you. That's all for today's video.